Okay, here's a fun one for you. What do your hair, the oxygen running through your blood, and yeah, even the milk in your fridge all have in common? It's a pretty wild mix, right? But the answer is actually a single type of molecule, one that basically acts as the microscopic engine of life itself. Yep, the answer is proteins. And in this explainer, we're going to dive deep into them. Because these aren't just some random molecules. They are the microscopic workhorses that are, and I mean this quite literally, responsible for just about everything that happens inside of you. So, to really get a handle on these workhorses, we first have to appreciate just how, well, fundamental they are. We're not talking about just one type of molecule among many. Nope, we're talking about the master molecules of life. Now, check this out. If you were to take out all the water in your body, about half of what's left over, half of your dry weight, is protein. That's more than anything else. From the muscles that let you move to the enzymes that digest your food, they are the essential substance of what makes you, well, you. Okay, so the big question is, how can one single type of molecule do so many wildly different things? Well, it turns out all of this incredible diversity, all this complexity, it all starts with a surprisingly simple set of building blocks. It's kind of like an alphabet for building life itself. And those building blocks, they're called amino acids. These are our letters. And just like with the alphabet, by linking these amino acids together in different orders, you can spell out all the different words, or in this case, proteins, each with its own totally unique job. So, if you look at this diagram, you'll see that all amino acids have the same basic backbone. It's like how all the letters in the alphabet might be written with the same pen. But the real key, the thing that makes each one different, is that part labeled R. That's the side chain. You can think of that as the unique shape of each letter. That R group is what gives every single amino acid its own special chemical personality. So, how many of these unique letters do you think are in life's alphabet? 100? 1,000? Nope. Just 20. Isn't that incredible? With just these 20 common amino acids, nature can build a practically infinite variety of proteins. It's the ultimate example of doing more with less. All right, so we've got our letters, our amino acids, and we've put them in a specific order. But how does a simple string of these things actually become a complex, working machine? Well, that's where the real magic happens. Folding. A protein isn't just a chain, it's a piece of molecular origami, a beautifully folded 3D structure. And this folding process doesn't just happen all at once. It's actually organized into 4 distinct levels. So let's do this. We're going to build a protein from the ground up, walking through each stage one by one. Okay, level 1, the primary structure. This one's easy. It's just the unique linear sequence of the amino acids. Think of it exactly like beads on a string. And that specific order, it's not random. It's spelled out, letter for letter, by the instructions written in your genes in your DNA. Right, but that string doesn't stay straight for long. Now we get to the secondary level. Here, little hydrogen bonds start pulling parts of the chain into regular repeating patterns. And you can see the 2 most common ones right here. You've got this cool spiral staircase shape called an alpha helix on the left, and this kind of folded paper or accordion shape called a beta-pleated sheet on the right. These are the first folds. Next up is the tertiary level, and honestly, this is the game changer. This is where the entire chain with all its little spirals and sheets folds up on itself. It scrunches up into a really complex and specific 3D shape. And this is the moment of truth. This is the point where the protein actually becomes a functional working machine. And then for some proteins, there's one final level, the quaternary structure. You can think of this as the team-up phase. This is where multiple already folded protein chains, we call them subunits, come together to form one giant functional complex. The hemoglobin molecule you see here, the one that carries oxygen in your blood, it's a perfect example. It's actually made of 4 separate protein subunits all working together in perfect sync. Okay, so we have these incredibly intricate 3D shapes. But what do they actually do? What's their purpose? Well, it all comes down to one simple rule. Their job description is determined entirely by their shape. And look, if you remember just one thing from this entire explainer, make it this. For a protein, structure is function. The shape is everything. That unique 3D fold is precisely what allows it to do all these amazing jobs in the body. Things like being structural support, transporting stuff around, speeding up chemical reactions, creating movement, and even defending us from germs. Let's take a look at a great example. Collagen. Its whole job is to provide structural support. And how does it do it? You can see its shape here. 3 chains wind around each other to form this super strong rope-like triple helix. It's this exact shape that gives our skin its elasticity and our bones and tendons their incredible strength. Shape equals function. And let's circle back to our friend hemoglobin, the oxygen transporter. This graph shows just how brilliant its design is. See, in the lungs, where there's a ton of oxygen, hemoglobin's shape allows it to grab onto oxygen molecules really tightly. But then, when it travels to your muscles and tissues where oxygen is needed, its shape changes ever so slightly and it lets go of the oxygen, releasing it right where it's needed most. It's an absolutely perfect delivery system. So we've established over and over that a protein's specific shape is absolutely critical for its job. This leads to a really important conclusion. If you mess with the shape, you completely mess with its ability to work. So what happens when proteins go wrong? Well, this loss of shape has a name. It's called denaturation. Things like extreme heat or changes in pH can cause a protein to completely unravel and lose its 3D structure. You've actually seen this happen a million times. When you fry an egg, the heat is denaturing the proteins in the egg white. They go from that clear, runny liquid to a solid white. The protein is still there, sure, but it's just a tangled mess. It's completely lost its function. And this image shows it perfectly. On the left, you have the native protein, all perfectly folded and ready to work. But after you denature it, you get what's on the right, a useless, unfolded tangle. Now, the important thing to realize is that the primary sequence, the string of amino acids, is still intact. All the letters are there. But without that specific 3D fold, the protein is essentially broken. But sometimes the problem isn't about unfolding the whole protein. Sometimes the problem is way, way smaller, but the consequences can be just as staggering. Take sickle cell anemia. This entire disease is caused by a change in just one single amino acid in the hemoglobin protein, out of hundreds. As you can see, a single glutamic acid gets swapped out for a valine. That's it, just one tiny typo in the genetic code. And just look at the devastating result of that one-letter change. That tiny swap causes the faulty hemoglobin molecules to start clumping together, forming these long, rigid fibers inside the red blood cells. This forces the cells, which should be nice and round, into the stiff sickle shape you see here. And these sickled cells, they get stuck in tiny blood vessels, blocking blood flow, causing incredible pain and organ damage. And it all goes back to that single tiny error in the amino acid sequence. It's just an incredible story when you think about it. We go from a simple 20-letter alphabet all the way to the complex 3-dimensional machinery of life. But it's also a system that demands incredible precision, where we've seen the smallest possible change can have the biggest, most profound impact. Which really makes you wonder, right? It leaves us with one final big question. If one single letter can completely change the story, what other secrets are our proteins still waiting to tell us?